they are getting deeper and deeper as the abundance of both whales and also sea lions begins to decline. And so we notice they finally get to move to their preferred habitat as the whales depart. And interestingly, the sea lions follow suit. Sea lions are like, they're like raccoons, scavengers, if you will. They like to cheat, they like to save energy, and they just are plain annoying. Kind of like this. So when you see the whales doing their thing, disrupting the herring and foraging efficiently, you also get the water dogs that are chasing them around. All right, and so this is a conspicuous behavior, and happens, we think, only let me rephrase that. The whales are providing opportunities for efficient sea lion foraging. Sea lions can go all the way down to the depth. There's no problem with that. They can deal with that, but it's much more efficient to only go half the depth, if you will, to go down to 50, 60, 70 meters versus 150. And so the whales are keeping the herring up, we think, and the sea lions are making use of that. And so here's what this looks like. It sucks to be a herring pretty much in the wintertime. So this is that same imaging sonar, but we've deployed it inside the layers. So this is kind of an example of what it would look like, where the layers would be here. We've got the Ditson looking at them, and the sea lions, of course, are coming and doing their thing. So this is about 50 meters, which puts this at about 60 or so meters. So it's really good for the sea lions. The whales are there distributing them, disturbing them, making them basically school and be very easy prey for these other predators. So we don't really know necessarily what the cumulative effect is of this, but we hypothesize that, that because the whales are present, it's also influencing the sea lions' ability to forage, and also makes them available to other predators like seabirds and other piscivores, which can make use of them. I mentioned earlier that we're continuing to carry on these observations because the populations in Southeast Alaska of herring have not really been doing so well lately, and I think that it's related to a meteorological phenomena. The last 3 years, two of the last 3 years of herring haven't shown up. They didn't come back, which is weird. And then this last year, they have shown up, but they've shown up in less abundance than they had been in years past. So it's unclear what that will ultimately result in with respect to the fate of the population of herring. But we know that the whales are also doing something different, which is really interesting. And so we have a student, she's actually graduated now, out of Southeast Alaska, and her job was to go and think about what are whales doing now that herring are less abundant. How are they making a living? And so some of them have found a way to efficiently feed at salmon hatcheries. And so this is a humpback whale right next to the pen of the salmon hatchery. And we notice that there's a few individuals that seem to be displaying this behavior. So it's not that they're not able to make a living, but they may be doing it in different ways. And so perhaps they're taking advantage of people's efforts for restocking, in this case, salmon. And whales are now finding efficient ways to forage on all of that effort. So what happens is they take the net pins out, they release the salmon, and then the whales, of course, are just like, hey, free lunch. And so some of them have done this, and it's interesting because it's the same. It seems to be the same individuals that are doing this and have been doing it for the last couple of years. So Ellen, she's the PhD student who worked on this, just graduated, and has done some really interesting work with that. And thinking about what it costs, what is the energetic cost for lunging for these types of organisms versus krill or versus herring. And it turns out that they don't really make a great living. They're about breaking even here, whereas if they're going for some of the other prey, they may actually be losing energy at the end of the day. All right, so that gets us that was sort of the middle or intermediate scale. And now we're moving to the broadest scale of things that we've been working on. And it brings us back to the Gulf of Mexico. This is a project funded by the Gulf of Mexico Research Initiative, part of the consortium called DeepEnd, which several folks here are a part of, and many others, which this is only a handful of the organizations involved. But this is a large multidisciplinary program that we've been involved in for the last few years and stemmed from, of course, unfortunately, the oil spill from 2010. And so it follows on the effort that came from that as part of the NERDA response. And so in that NERDA response, we had a suite of stations, as can seen here, that were occupied almost continuously during that 2010-2011 period for about a year's duration following the spill, and resulted in a tremendous amount of data that we collected, and of course many other people have been involved in this as well, and they've collected all kinds of information. For us, it's been over accumulation of 1,000 net tows, lots and lots of acoustic data, lots of time at sea, and lots of new discoveries. And one of those discoveries, well, I'll get to that, how we're doing this. The main approaches that I'll discuss for the rest of this are the two sampling approaches here, the MOCNES, which is a multiple opening closing net environmental sampling system, sensing system, I think, the MOCNES. Anyway, it's a multi-net system, so it's a big frame, it's a 10-meter square net. And so we drop it down, and it goes down with the net open all the way down to the bottom. This is the footprint of that. Closes, opens up a net, closes, opens up a net, and so forth. And so we get depth discrete data, which we can then compare directly with our acoustics. And so we have multi-frequency acoustic systems, 18, 38, 70, and 120 kilohertz systems that we've been using. And we use them simultaneously to try to characterize what is in the water column. And so it's an interesting thing to be able to try to put these things together. But one of the pieces that we've been really interested in focusing on has been thinking about scattering layers. And so these are the deep scattering layers that exist throughout the world's oceans. Okay, so I'll introduce you to this figure. So this is water depth, and this is time. And so what you see is a color picture, which is, we call it an echogram. This is intensity, so more stuff here, less stuff here, and it changes in depth across time. All right, so these are called deep scattering layers, sound scattering layers, lots of things like this. So what you see here, the prevalent behavior is something called diel vertical migration, which you probably all know about. But what I want to bring your attention to is how variable this pattern or process is across the world's oceans. So it's ubiquitous, it happens across all the oceans, but it doesn't necessarily happen the same way, which I think is the really interesting part. And that's kind of what's led a lot of the stuff we've done for the Deep End project, and then going back to the data we collected for the NERDA program. So what we do notice is that these are organized in these sort of depth discrete layers, right? And so at nighttime, they're up in the water column, presumably foraging, and then they move down at depth during the daytime, and then they come back up at night. And so we notice again that the structure is a little bit different across the different world's oceans. The Gulf of Mexico isn't here, but I will show you something similar. This is important, of course, because of this process here. So carbon transport. And so all this material moving vertically hundreds of meters a day. The world's largest animal migration happens twice a day. Okay? World's largest animal migration on the world, globe thing, happens twice a day, right? So a tremendous amount of materials being transferred from the mesopelagic to the upper water column, or the EPI Pelagic. And that's what I think is the part we're really interested in. We have one of these things in the Gulf of Mexico, this spirally looking toilet bowl thing, the loop current, and then the derivative eddies that come off of it. And so occasionally we end up sampling in some of this area. And one of the things that we're interested in trying to understand is, how does this feature, this important dominant feature, how does it drive or characterize the scattering layers that we see? So I showed you that across the world's oceans there's variation. And we're beginning to pick up hints that the variation may also be, there may be variation within the Gulf across the areas that we've sampled, and likely we think is a function of this. So this is something that we're working on now. So the results are limited, but I think they open up some opportunity to have some interesting conversations about what may be driving this. So the next slide, I'll show comparisons between two dominant features. We have a cyclone and anticyclone. Generally, cyclonic systems are considered to be high productivity. Anticyclonic systems are generally believed to be low productivity, right? So you have an upwelling system or a downwelling system, and the associated productivity with that. So The presumption then is, of course, if you're in the Gulf of Mexico, you're in the anticyclonic system, you're likely to see reduced productivity or biomass in comparison to the cyclonic system. So we first look at the EPI Pelagic Zone, so that's a 0 to 200 meter. And we then look at this metric called SA. So this is logarithmic scale. This is in decibels. I'll draw your attention to the differences. These are important. So a 3 dB difference is a factor of 2. A 10 dB difference is a order of magnitude difference. All right, so small variation or small differences actually yield meaningful results. So we have the cyclonic region and the anticyclonic region here. And as you might imagine, between day and night, there's a difference. And the reason is because of this diel vertical migration. I just explained that. So during the daytime, things are generally low in the epipelagic, biomass levels, if you will. During the nighttime, they increase, right? And they increase by quite a bit, almost 10 dB here, right? So what this suggests is that during the daytime, there's not as much material in the epipelagic as there is in the nighttime, which is good. In the mesopelagic, we see something, I guess, similar, you'd expect. During the daytime, it would be a little bit higher. During the nighttime, it would be a little bit lower because those organisms are moving from the mesopelagic into the epipelagic. But what we see that wasn't expected is that the cyclonic regions, both day and night, tend to have a lower biomass, if you will, than the anticyclonic regions, either day or night. And so we expected that the cyclonic regions would be more productive, have more biomass than the anticyclonic systems. And it's suggesting that maybe that's not the case. Maybe there's some other factor that's mediating this. But if we just took everything and combined it all into a single number, which is shown here, this is the result we get. With help from Schwanman and his group, we've been able to look a little bit further into this, thinking about gradients of these responses. This is one panel of a multi-panel graph. I didn't bring it or didn't show it just to kind of reduce complexity here. But this is, if you're in an anticyclonic system, the AR index, this intensity index, it's a how extreme, if you will, of an anticyclonic system are you in. And so I'm not a physical oceanographer, so if you guys are and I offend you, I apologize. But this suggests that it's more intense downwelling than here, and then we would move into the cyclonic and then common water areas below that. And so what we show here is solar elevation, so basically a function of light levels, which we know is important for structuring the movement. The periodicity in these layers moving in the water column is generally shown to be a function of light level. So light intensity is important, and they're believed to follow isolooms. So these organisms will orient to particular isolooms and move vertically through the water column in association with that. But what we see is that, interestingly, the gradient here, as a function of the intensity that you're in. And so as a function of daytime, basically night and day in the epipelagic, you don't expect much there, right, because they're in the mesopelagic. But at nighttime, they will move up, and they move up, but they don't necessarily do it evenly. So there is physical forcing, we think, that exists here. Again, this is something that's kind of new. We're still sorting out how to best interpret this, but it suggests that, I think at least, that some of the physics might be dominating how these organisms are concentrated, and in a way that we can actually measure them acoustically. This is not a great way to demonstrate light intensity, and like I mentioned, Schwanman's group is helping us kind of think about this, where we can actually discretely measure light intensity in the water column. So the mesopelagic one is one that's still kind of baffling with regard to this gradient. I would expect, I guess I'm a little surprised that we see highest peaks in biomass associated with the highest intensity of these physical features. So if anyone has any comments or thoughts on this, we're still trying to wrap our heads around what may be driving this. It'd be great to discuss this more. So this is the process in the Gulf of Mexico: nighttime, daytime, nighttime. But if you look closely, and hopefully you can see it, it's not just one mass of things moving. There's filaments, there's pieces that comprise this behavior. So if you look closely, you can see there's a dominant layer there, there's another one there, and then they go to different depths, which meaning that they're migrating different distances. So first, second, and third. So if you squint, you can see 3 layers in here. the first layer actually goes further than the second and third. So they're moving at different times, and they're moving at different distances, which suggests they're also moving at different rates. Right? So these different layers are comprised of different organisms that are queuing into something which we're still trying to piece together, possibly light, possibly each other, possibly sound, maybe chemical cues. But it's opening up some really interesting questions about how these things are structured in the Gulf of Mexico and what may be mediating these movements of these layers from the surface to the bottom and then back up.
The 3 layers you're referring to, I'm having a hard time.
Oh, you can't see that? Come on, dude.
I see maybe 6 or 7, so I wasn't sure.
Oh, well, you know, this is a bad figure. So, yeah, so there's one here, one. Oh, here's a pointer. I'll use them both. How about that? Is that better? Yeah. All right, so we got one here, one there, and one there. And over here, in most cases, so I guess I should qualify this statement. In 92% of the cases, there were 3. Sometimes there were 4. And there aren't necessarily always 3 going down and 3 going back up. So it's a bit more complicated than just saying there's 3 layers. But in this case, if I were to tell you which one this graph represents, I'd say, or well, this is all of them together. But in this figure, if I were identifying them, there's one there, one there, one there, and possibly the start of one that sort of dissipates. And here, you'll notice that when they go up, there's actually multiple groups that may be originating and moving up. But on average, the majority of the time, there were 3 dominant layers that moved. And so the fact that in one station or one location you see this, lots of complexity, and in another one, it's much more simple, again, is another interesting feature of this that begins to, well, we need to ask, well, what's dominating this or what's driving this variation? And that's the part that I think we're most interested in now in spending our effort trying to tease apart. So in trying to answer this question, we now think about using the acoustic data to separate into relevant taxonomic groups. And we do that by examining the acoustic responses. So we have multiple frequencies, and each frequency generates a response. And so we can compare one response to another in the same volume and begin to think about how an organism would respond to sound across these different frequencies, and then begin to think about, based on models that we have, which category that organism might fit into. And so we can broadly classify acoustic responses based on something called like dB differencing, where we're basically looking at the difference between one frequency and another frequency and recognizing how these organisms would respond to sound and classify them to these groups. And so just as an example, 2 different migrations, you can see that there's inherent complexity. All other things being equal, right? The responses across these are quite striking. And the depths themselves are also different, which is very interesting. The dominant patterns of these layers, and also the ones that don't move, right? So not everything migrates vertically. A lot of it does, but there's also a resident population or non-migrators that may migrate, once every 3rd day or something like that. So it's offering some opportunities to kind of think about this, and then to begin to understand, well, what proportion of these is comprising any of these layers that we see, right? Which of these groups, these taxonomic groups, contributes most to one of those? And then more importantly, I think, gets us into asking questions about carbon, right? How is carbon being moved from the mesopelagic into the epipelagic? And which groups are most important in structuring or driving that? And so you can see, I just highlighted this again, these are the 2. So on the downward phase, we find that those with gas inclusions, and so that's going to be fish with swim bladders and other invertebrates that have gas inclusions, their contribution is 0.8 to 1.2 milligrams of carbon per square meter per day. This is, I should qualify this again, back of the envelope calculation here, which we all like to do occasionally, but it demonstrates some interesting patterns, again, all other things being equal, that the large non-swim bladdered fish, so these are fish, yay size, that don't have swim bladders, so they don't scatter sound in the same way. But there are many of them, are an order of magnitude more in terms of the carbon flux that's attributed to that group. And then on the upward phase, we see something similar for those 2 groups. So this is just an example, and this is the pathway which our group is leading toward. if anyone's interested in modeling carbon flux and trying to think about those gains and losses, I would really be interested to talk to you, because we can get it to here with the acoustics, but broader scale sort of implications would be really nice to consider. Oh, try that again. Helps when you go the right way. Okay, so that's all cool, and all of that I've shown you comes from things that look like this. So you have a ship, you have these sonars, and they're sending out sound and receiving sound and integrating large volumes of water, right? So which means that the responses that you're seeing come from a lot of integrated volume and lots of different organisms contributing to that signal that you get. And so that's nice, and it helps to describe large-scale patterns, but it doesn't help us really get to the crux of the questions that I've mentioned I'm interested in addressing. And so that's led us to move beyond the ship. This is still important, we still do this, but then taking these instruments, as shown here, and sticking them down at depth into the domains where these organisms actually exist, and thinking about the small scale or fine scale patterns. So this is an example of one evening. We've got the layers here. This is the Mach Ness path, which we've sort of overlaid onto this. And on the Mach Ness has our sensors. And so the sensors go through all of these. And the sensors are only looking out to about 10 meters or maybe 20 meters. And so the closer you get to an organism with these devices, the greater your resolution, which means that we can now see individuals. So we don't necessarily see this anymore. We still see this from the ship, but the sonar that's deployed inside this, that goes all the way through it, right, that goes through this whole slug, provides us something that's much more interesting. And so these are now individuals. Okay, so we've gone from the aggregate to the individual at depth, and we have this all the way through. So even when we get down to here, where there are still organisms, we can compute in situ densities. We can then think about how these might relate to one another. We have the ability, whoa, I gave it away. Have the ability to track them. And so this is the behavioral component now. We can start to think about how do these individuals relate to one another? How do they respond to the physics? How do they change over time and as a function of depth? And so we can look at those like we would any other acoustic approach, but we can do it at depth, which is really informative. And then with the new approaches or new technologies that we're using, our broadband or wideband techniques, we can now look at the acoustic response. So this is frequency. You don't need to see the frequencies, but just this is intensity, and this is a suite of frequencies, or a spectra, if you will. And so these instruments have the ability to collect data not just at a single frequency, but think of it like a full spectrum. And so kind of like a GC or a mass spec, right, would provide you these signatures. We get acoustic fingerprints now of these organisms. And so what this allows us to do then is to now look at these individuals as single particles, look at their acoustic fingerprint, if you will, and then begin to tease apart who's really contributing to the volume, or this volume scattering that we see, and how does Bob over here, who's next to Susie, how are they responding? And are they the same organism, or are they different organisms? And then ultimately, of course, thinking about if we can get to the individual